Hello, everybody. I'd like to welcome you to the 22nd session of the GO2NE Global Ocean Oxygen Network webinar series. I'm Lisa Levin from the Scripps Institution of Oceanography at UC San Diego in the USA. And the whole GO2NE team is very happy that you took the time to be with us and our incredible speakers this morning, afternoon, and evening. Advance here. My next slide is not happening. There we go. Before we start, I'd like to bring your attention to the Ocean Deoxygenation newsletter, Good News, which you can access via the QR code you see here. If you are on Twitter, you can also follow us at good_oxygen_oxy for the latest news and activities of the GO2NE community. The next GO2NE webinar will be on October 11th, so please look out for details to register when the webinar is announced. I have some quick housekeeping announcements before we welcome our speakers. You as attendees have the possibility to type your questions in the Q&A box in Zoom, so please use that box. The chat can be used to share additional information or if you have any logistical problems, but please put the questions into the Q&A. So let's get started. We have two great scientists from Norway and the University of Bergen today. And although they will be speaking about other parts of the world, not just Norway. So these are Natalya Gallo, who will speak first, followed by Anna Groh. Both speakers have 20 minutes to present, and each will be followed by 10 minutes or 8 minutes of Q&A. So I'd like to introduce Natalya first. Natalya is a marine ecologist and biological oceanographer in the Fjord and Coastal Ecology Research Group at the Department of Biological Sciences at the University of Bergen, and she's an affiliate of the Bjerknes Center for Climate Research. She has a longstanding interest in ocean deoxygenation and has studied how this affects fishes in the California margin, in the Gulf of California and Mexico, and in the West Norwegian fjords. Natalya is also now a member of the GO2NE Steering Committee, new member. Today, she will present on the trophic impacts of deoxygenation, disruption of bentho-pelagic coupling in oxygen minimum zones. So I will turn it over to Natalya now.
Thank you very much for the introduction, Lisa, and thank you to everyone who's taken the time to join us today. So hopefully my presentation is showing now. Okay, great. So this is actually work I'm presenting that was done when I was a PhD student with Lisa at the Scripps Institution of Oceanography. And I think this was one of the most exciting findings from my PhD. And I'm still working on publishing it. And so it's a great opportunity to share it with you today because I think that oxygen minimum zones play a really important role in shaping the patterns of demersal fish that we see around the world through this important bentho-pelagic coupling process. But since this is a webinar about ocean deoxygenation, I first wanted to start by mentioning that the reason why it's important to understand how low oxygen environments affect marine ecology and ecosystem productivity is because right now we're seeing large changes in ocean oxygen. So over 50 years between 1960 and 2010, there was a reduction in the global ocean oxygen inventory by about 2%. And this occurred both in shallow waters as well as in deep waters. And it's important to note that the observed decline in oxygen has actually occurred faster than what climate models have predicted would have happened during that time. So while there's been a 2% decline in the observed oxygen during the same period, the models have only predicted that that decline would have been about 0.6%. And going further into the future under different global warming scenarios, we see even larger losses in global oxygen levels predicted. And so one of the questions I'm really interested in is what this means for ocean ecosystems, and specifically for demersal fish and fisheries. So demersal fish are fish that are associated with the seafloor. Typically, they're fished with bottom trawls. There are really important demersal fisheries all around the world. Some examples of demersal fish that you might be familiar with include things like hake. Rockfish, for example, are very common on the US West Coast. Orange roughy was a really important deep sea demersal fish that was overfished. Cod, of course, is a very important demersal fish. Pollock, saithe here in the North Atlantic are important. And then there's halibut, sablefish, cusk. There's a whole variety of different demersal fish that you might find at your local market where you might go to buy fish. And one of the questions I'm really interested in is how low oxygen environments affect the trophic relationships that demersal fish have. Basically, who the demersal fish are feeding on, what organisms. And so here we're taking a look at an oxygen profile taken in the Southern California Bight, where the study took place. And so basically, if you used a CTD and lowered it through the water column, off the coast of California, you would encounter these different water masses that have different oxygen properties. So just a little shallower than 300 meters, you would actually encounter hypoxic waters. And this region is considered to be the oxygen limited zone. And this is an area where you start seeing a change in the composition of the demersal fish community based on which members are more or less tolerant to hypoxic conditions, hypoxia being low oxygen conditions. This is basically where the conditions first start being physiologically challenging for certain species. As you move deeper in the water column by around 450 meters, you encounter a zone known as the oxygen minimum zone. And here conditions are severely hypoxic. So you can see that the oxygen levels are much lower. And here really it's just the very hypoxia-tolerant demersal fish species that are able to inhabit this zone. And as you move deeper, oxygen levels actually increase and you move outside of the oxygen minimum zone, but my study doesn't really sample below the oxygen minimum zone in the Southern California Bight. And my study here and my talk today is really focused on this phenomenon of bentho-pelagic coupling. And so bentho-pelagic coupling basically means the way different nutrients or energy or mass is connected between the shallow surface waters of the ocean and the deeper seafloor regions of the ocean. And there are many different processes that contribute to bentho-pelagic coupling, but the one that I'll speak about today is this active form of bentho-pelagic coupling, which is mediated through diel vertically migrating organisms. And so these are organisms, they're, for example, mesopelagic fish, krill, copepods that are found deep during the day, where they're basically trying to be in a region of low light. And then they migrate up at night and they feed on the regions of the ocean in the shallow water where there's a lot of primary productivity. So they're feeding on the phytoplankton up there. And these organisms perform this migration every single day. And the component of this migration that I'm really interested in is what happens when this diel vertical migrating community actually intercepts the seafloor because all of a sudden you're in an area of shallow topography. Or what happens when this community intercepts a low oxygen water mass, which can also act, as you'll see, like a false bottom. But basically, these diel vertically migrating organisms are really interesting from the perspective of the demersal fish community because they can provide a really important nutrient subsidy. Basically, they're feeding at the surface, swimming down, intercepting the seafloor, and then all of a sudden you have all of these demersal fish on the seafloor that get this very easy prey resource that's come down from the surface. And so I consider this to be a demersal fish mediated bentho-pelagic coupling, which is what I'll be speaking about today. And you can think about this as kind of a seafloor of mouths. And we see this at play on the California margin. So here I'm just showing you a frame from an ROV video. And the ROV was right near the seafloor at a depth of about 340 meters. And this is a region of the Southern California Bight that we had been studying, and that's why we were trying to do an ROV dive there. but we could see this really high biomass of krill found right above the bottom. And we can see it on the echo sounders too. So here at the bottom, you can see an image that I just grabbed from the echo sounder on the ship. And you can see this really high backscatter right by the seafloor at 340 meters. That's basically these diel vertical migrators during the day being compressed and intercepting the seafloor. And when you look at the fish community, the demersal fish community in this area, and you look at what species are present and what they're eating, what you find is that the fish community in this location is very much composed of fish that feed on this prey because it's a very abundant, easy to access prey resource. So just to reiterate, in areas where the diel vertical migrators come down to the bottom, you find a fish community that's dominated by pelagic feeders. And this is consistent. This has been described for other demersal fish communities around the world, if, for example, in areas in the North Atlantic by other authors as well. But we know that oxygen minimum zones can affect the daytime depth of the diel vertical migrating community. And so that's what this figure from Bianchi et al. Shows, that basically in regions where you have low oxygen zones, that daytime depth of the diel vertical migrating community is shallower than in regions that are well oxygenated and don't have oxygen minimum zones. So basically these OMZs, they compress that daytime depth of the DVM community. And so this I'm just showing you, this is where our study takes place in the Southern California Bight. And this is data specifically from the Southern California Bight from our colleagues, and they found that the upper and lower boundary of the deep scattering layer were significantly correlated with oxygen thresholds in the Southern California Bight. So that leads to my research question. Is this demersal fish-mediated bentho-pelagic coupling impaired by oxygen limitation in oxygen minimum zone regions? And so I just want to point out that demersal fish can feed on benthic or pelagic prey. However, the pelagic pathway has greater trophic efficiency and therefore can support a higher fish biomass than the benthic and detrital pathway. So if you look at this illustration on the right, basically you have primary production, pelagic organisms. These are these diel vertically migrating organisms like krill or mesopelagic fish. And so you can either be a fish that feeds directly on this, or you can be a fish that feeds on those organisms as they die and fall down to the benthos and get decomposed or eaten by benthic organisms and then feed on those benthic organisms. So basically the benthic food pathway is a longer food chain, and therefore it's less efficient. And so the way we're going to look at this loss of bentho-pelagic coupling is we're going to look at the changes in degree of pelagic feeding, the trophic level of the fish that are present, and the trophic diversity. And so for this study, we conducted 38 trawls in the Southern California Bight, ranging from depths of 96 to 1261 meters. And these depths correspond to these different oxygen zones. So basically, depths that were shallower than 450 meters, we characterize as part of the non-oxygen minimum zone demersal fish community. Trawls within 450 meters and deeper are part of the oxygen minimum zone. And then the trawls that are between 700 and about 850 meters are this oxygen minimum zone core community where the oxygen levels are lowest. And again, just to reiterate, these are all demersal fish. So these trawls are done on the seafloor at these different depths. And we determined the community composition for each trawl in order to look at which species were present and in which relative abundances. And then we also conducted a lot of different types of diet analyses. So our two main methodologies were gut content analysis and stable isotope analysis. That's because these are two different methods that can give you a good snapshot of the trophic ecology of the species of interest. So for the gut content analysis, we identified all of the gut contents to the lowest possible taxa. And also we calculated Levin standardized niche breadth and Shannon-Wiener diversity index for the diet contents. Based on the diet contents, we also assigned a benthic or pelagic feeding mode to every single species that was part of our community. And we also looked at any additional references that had information about the diets of all of our species in order to make that assignment. And then on top of that, from each trawl, we would sample about 3 individuals from each species to run for stable isotope analysis. So we used white muscle tissue for stable isotope analysis and ran it for carbon-13 and nitrogen-15. And we applied a lipid correction for the samples that had a high carbon to nitrogen ratio. The reason why we used stable isotope analysis is because, unlike diet analysis, which just gives you a snapshot of what the fish ate before it was captured, stable isotopes allow you to look at an integrated signature of what the fish was eating on for the previous few months. And so carbon allows us to look at what the base of the food chain is, and nitrogen allows us to look at how the species compare to each other in terms of which trophic level they're feeding on. And additionally, we used Lehman metrics, which are shown here, to look at how different the trophic niches of the different species were, as well as the trophic niche of the community as a whole in these different oxygen zones. So first, the first conclusion is that the demersal fish communities within the oxygen minimum zone core were distinct from those outside of the oxygen minimum zone. And so here you can just see some example communities from 200, 340, and 700 meters. And so you can see that the dominant species shifts as you move deeper into the oxygen minimum zone, and visually too. You can see that these are 2 very different types of fish communities. But it's not just that the species change, the dominant feeding mode changes as well. So when we look at what the proportion of benthic to pelagic feeders was for every single trawl, and so here the points represent every single trawl, all of the 37 trawls in our data set, you can see that when you're in shallower water, so the x-axis here is showing you depth, and so from left to right we move deeper. In the shallower trawls, we see a fish community that's dominated by pelagic feeders, whereas when we move deeper and into the oxygen minimum zone, we now find a fish community that's dominated by benthic feeders. When we look at this at the species level as well, we have certain species that are never present in the oxygen minimum zone. These are probably our intolerant species. We have species that occur both inside and outside of the oxygen minimum zone, some of our transitional species shown here in blue. And then we have the oxygen minimum zone specialists, which here are only found within the oxygen minimum zone, shown here in red. And here I've calculated the trophic level based on the background particulate organic matter signature and using +3 per mil enrichment to estimate an additional trophic level. And what we can see is that the OMZ specialists are feeding at a higher trophic level than both the hypoxia-intolerant species outside of the OMZ, but also compared to some of these transitional species. We also can see that the oxygen minimum zone core community has a narrower trophic niche than the non-oxygen minimum zone community. So you see an enrichment in nitrogen in that oxygen minimum zone community, but you also see an overall decrease in the trophic niche. And partially this is driven by the fact that there are fewer species, demersal fish species, living in the oxygen minimum zone, but also fewer prey resources that they're utilizing. Generally, we see very little change in the carbon signatures across these different oxygen zones. But again, as I've said, a strong enrichment in nitrogen, which is a marker for trophic level. And we see that this enrichment in nitrogen really appears to be correlated, but in a nonlinear way with oxygen. So here in this range within the oxygen minimum zone, we see this strong enrichment in nitrogen in the demersal fish muscle signatures across this oxygen gradient here. And if we go back to our finding that we see this transition from a pelagic feeding community to a benthic feeding community, this actually corresponds very nicely with the change in the mean delta N15 signature. So basically, as the fish community changes from one that's feeding primarily on pelagic prey to one that's feeding primarily on benthic prey, that coincides with this overall increase in the nitrogen signature of the demersal fish that are present in that community. So basically, that's likely what's driving that enrichment in nitrogen 15 in the OMZ. And this matters because in the Southern California bight, only 20% of the seafloor is present at depths that are shallower than the upper boundary of the oxygen minimum zone. And so basically, it's this very small fraction of the seafloor in the demersal fish communities in the Southern California bight that can access these pelagic organisms. And this contrasts with what's happening on well-oxygenated margins. So this figure is from a study done in the Northeast Atlantic. And here, the diel vertical migrators supported demersal fish down to depths of 1,500 meters. And so we see this transition at 450 meters from a pelagic supported to a benthic supported community. But if not for the presence of an oxygen minimum zone, this diel vertical migrating prey subsidy could go much, much deeper. And so the oxygen minimum zone is basically compressing this. And this is quite important to think about because, first of all, when we're thinking about global patterns of demersal fish, it's important to remember this linkage with pelagic prey. And this is often left out of ecosystem modeling studies. So this, for example, as a great study, but for the demersal fish, they assume that only demersal fish at depths shallower than 200 meters have any interaction with pelagic prey directly, whereas anything that's deeper than 200 meters is only utilizing benthic resources, which is not what's happening. And so it could contribute to differences in these global findings. And one of the questions that comes up is this reduced bentho-pelagic coupling an underlying ecological property of other hypoxic marine systems, such as fjords and closed seas and estuaries? And so this is one of the questions that we're looking at right now in the Hippon Fjordfish project here in Norway. And so just to conclude, one, high biomass demersal fish species prey on diel vertical migrators, and this feeding interaction is an important bentho-pelagic coupling process. We see that oxygen minimum zones compress the daytime distribution of the DVM community, leading to a reduction in bentho-pelagic coupling on oxygen minimum zone margins. Fishes that live within the oxygen minimum zone are predominantly benthic feeders and feed at a higher trophic level than fishes above the oxygen minimum zone. And deoxygenation and oxygen minimum zone expansion may lead to a more benthic-associated food web and impact the productivity of West Coast demersal fisheries in the future. And with that, I would just like to thank the many students who contributed to this analysis, especially for the gut content and stable isotope work, and open it for questions. Thank you.
Thank you, Natalya, for that really excellent presentation. And I would encourage anybody with questions to put them in the Q&A. There is one question in the Q&A that's not specific to your talk, but I actually have a question that is specific to your talk, so I think I'm going to ask you that first. It looks like hypoxic waters between 63 and 22 micromolar of oxygen are included in your non-OMZ category. And we know in coastal waters that oxygen at that level has a really big impact on communities. So I wondered if you see any changes associated with either composition or feeding behaviors at that level of, say, 63 to 22 micromolar.
Yeah, so I mean, we do see the transition with the nitrogen enrichment, and here I will just quickly share this slide. We don't see that it's completely an abrupt transition right at the upper boundary of the oxygen minimum zone. It is a gradual transition that does start to occur under hypoxic conditions within the oxygen limited zone. And I believe that for other systems where kind of the background history of the oxygen environment is different than the Eastern Pacific, which has very large low oxygen areas, you would probably start seeing this shift occurring at higher oxygen levels. So basically the species there would likely be more sensitive to to higher levels of oxygen. Basically, the hypoxia thresholds would not be as low there as they are here in the Eastern Pacific. And so I think for every system, you would have to separately look at where these shifts start to take place. But one question I leave the audience with is, how common is this pattern? Do we see these decreases in bentho-pelagic coupling across hypoxic systems, shallow ones that are not nearly as hypoxic as here in the Eastern Pacific?
Great, thank you. We have a question from Ahmed Mohammadpour, and it says, thank you for your nice presentation. I was wondering if demersal fishing can impose any impacts on the OMZ as well.
Yeah, that's an interesting question. I haven't thought about it. Certainly, off California, some of our demersal fisheries do fish species that live in the oxygen minimum zone. However, these tend to be less productive communities, so the biomass is lower, so I think the effort is a little bit lower in those areas. The only direct effect I could think of is maybe through like increased resuspension of sediment and potential remineralization, with organic matter being resuspended from like trawls, for example. But other than that, I can't really think of a clear linkage between how fishing would affect the oxygen minimum zone conditions directly.
Thank you. We have a message, a question from Christoph Rabui. It says, nice talk. How do you think that seasonal hypoxic conditions may influence demersal fish populations, such as in the northern Gulf of Mexico?
Thank you for the question. I imagine that seasonally you would see similar types of changes in the bentho-pelagic coupling. And so it may be these more episodic shifts. That's also what I suspect we see, not at seasonal, but kind of more at these interannual timescales in places like fjords that may have hypoxic conditions that occur from time to time. But yeah, there's also the possibility that in shallower systems, and potentially this is the case for areas in the Gulf of Mexico, where you could see, or areas that are even shallower, like the Chesapeake Bay, for example, where maybe you see more benthic coupling because, I don't know, because everything dies and all of a sudden the depth isn't so great. And so then the fish are coming down and feeding and then coming back up quickly in order to respire. So, I mean, it could be different in shallow water systems than deep water systems, but I imagine that in the Gulf of Mexico, where it's seasonal, it would be a similar relationship, but more episodic with the season. But certainly it could be looked at and would be really interesting to look at it across different systems.
Okay, we'll take one last question. Does the oxygen, let's see, from Godfrey Munoz, does the oxygen variability affect the marine ecosystem and thus result into distinctions of some species?
Yeah, thank you for the question. Certainly, I think variability comes into play, and we know that the upper boundary of the oxygen minimum zone and the oxygen limited zone isn't constant. So there's certainly variability that occurs at seasonal timescales and interannual and multi-decadal timescales, and also just a daily variability that's tidally associated. It probably contributes to more species being able to be these transitional species because they are able to be exposed to a larger range of oxygen and not just stable, very low oxygen conditions, even when within these otherwise hypoxic systems.
Okay, maybe I'll squeeze in one last question, but it'll have to be a fast answer from Parvati Valivattilam. Thank you for your interesting talk. Did you see any sensitivity to temperature and so are marine heat waves on the benthic interaction?
No, I didn't look at that, but I think it's an interesting area of research. And certainly we know that under warmer temperatures, the oxygen threshold at which you'll experience hypoxic stress will change. So basically under warmer conditions, animals respire more, they need more oxygen. So they would encounter more physiological stress of hypoxia under a heat wave condition.
Great. Well, thank you so much, Natalya. We're going to now move on to our next speaker. Any questions that people want to add in, they can, and we'll try to get them, answered offline to you. So our next speaker is Salvanes. She's a professor at the University of Bergen, Norway. Her main research field is in marine ecology and how the environment shapes fish behavior and ecosystem functioning. Her main study sites have been in the West Norwegian fjords and in the Benguela upwelling ecosystem. She works both in the field on board large research vessels, but also with experiments in aquaria. And today she will present on joint projects with local scientists from 20 years of studies of the hypoxia-tolerant bearded goby in the Namibian Benguela. And the title of her presentation is The Bearded Goby, a hypoxia-tolerant little fish member of the Namibian Benguela Ecosystem. So, Anna-Gro, it is all yours.
Thank you very much, Lisa. I try to share my screen now. Then also share my presentation. Yeah, see. So the goby is endemic to the Benguela. The shelf of Benguela is known for high productivity, high precipitation of organic material, and also low oxygen levels on the shelf area. Scientific survey data over 20 years demonstrate that the gobies are distributed over the shelf area along the Namibia. And when the economic zone was put to work in 1919, the gobies also came in as bycatch in the fisheries. Survey data has shown that gobies overlap with their predators, which are hake, horse mackerel, and also with the hypoxic water. And detailed analysis of those data has shown that the goby abundance decrease when the predator abundance increase, and also that the goby abundance increase where and when the oxygen levels decrease. Diet studies of the birds, seals, and also penguins and large fish have demonstrated that goby are important prey, as you can see here, and it's also come into the fisheries as mentioned. And we may wonder if overfishing made some space for the gobies. Catch statistics on hake and horse mackerel predators and also on sardines show that there were big overfishing of these fish. And that was due to improved fishing technology since the 1950s. There were large distant water fishing fleet that were fishing freely until the 1990s. And there was a lack of economic zones and lack of management. And as you can see, there is a big, back in the 1970s was the maximum population sizes or maximum catches, and then it just decreased afterwards. And we think, and gobies entered the landing statistics from the 1970s and after that, yeah. And that the bearded goby became a focus in marine research is actually due to a serendipitous discovery. And it was a diamond company, the De Beers, that explored the Benguela shelf for diamonds. And then they used ROVs and came over some footage with a small fish on the seabed. And they sent those footages to Professor Gibbons in South Africa, and he identified them to be bearded gobies on the seabed. And Professor Gibbons, he invited colleagues from the University of Bergen in Norway and also from the Institute of Marine Research, both in Namibia and Norway. And we got access to ship's time via the benefit program and also via the University of Bergen. And we all involved graduate students to do master projects on the goby ecology, life history, and distribution. And that's what I'm going to present some of the results that everybody has found during these years. So our team has study the goby's habitat, habits, and also life history. We have had massive field collections on board the research ships. We have used acoustics to locate where the pelagic organisms were. We have had depth stratified pelagic trawling with using what we call a multi-sampler that can open and close. They have 3 bags that can open and close at the decided depths. And we use CTDs to measure environmental variables like temperature, salinity, and oxygen. And we have had corals and grab to get hold of the sediments. And we also have had outdoor on deck then. We have had experiments where we have created extreme environments for the gobies by bubbling nitrogen gas into the aquaria or at the end or adding sulfides, which they do experience naturally in their environment. And then we have measured the response to reduce oxygen by quantifying changes in ventilation rate. And also we have measured the oxygen uptake by using respirometry. In addition, we have done controlled behavioral experiments on board the ship in the climate rooms, where we have looked into what kind of substrates do they choose, whether goby and horse mackerel responds similar or different to jellyfish. We also had experiments on that. And at Natmyrk, or on land, there were lab experiments. And well, gobies, they are in general, they are typically breed in nests, and the lab experiments then that was done then on land, they showed that they did reproduce in sites that were prepared by the males, and eggs were attached in the aquaria that was on the aquarium wall, and they hatched after 7 days when the temperature was 21 degrees in the lab, and probably takes a longer time if it's colder. And they also show that males cared for and ventilated the egg clutch. And they also followed the yolk sac larvae. They survived until the first feeding. But then we didn't have proper food for them, so they died, unfortunately. We've also been exploring a lot of field collections, or we, I say, and then it's the many master students have been looking at that. And to the left, you see, we show a diagram with the distance from the coast on the x-axis, and also the body weight of fish caught in the pelagic and in bottom trawls. What the data show was that the males tended to mostly breed at the seabed on the outer shelf area, 60 kilometers offshore. We didn't notice any males able to breed further inshore. And it also showed that males have 2 reproductive tactics that do differ in reproductive investment and size. It's the territorial males. They do have large body and long urogenital papilla. They have small testes and large seminal vesicles. The seminal vesicles are mucin-producing glands that they use to build their nests, and also to get the sperm to fit into the nest. And they make the nest, as I said, and they care for the egg clutches. And then we have these sneaker males. They have a small body, they have short papilla, and they mimic females. And they have large testes, but no glue, no very small seminal vesicles. And they do steal fertilization. So they mimic females and try to steal fertilizations in the air. And females also are small, and they have a shorter, blunt urogenitilla, and they eggs attached to the nest sites for the territorial males. So what our data also show, the acoustics data, is that gobies survive in what we call dead zones with poisonous gas. We found that little life exists in the first 20 to 60 meters above the bottom. That's shown with the white part on the acoustic diagram. But and there the oxygen level is very low. But we did catch gobies there, so they tolerated to be there. And what Acoustic and depth stratified trawl catches, they demonstrated diurnal vertical migration in the gobies. At night, they go shallower to where the oxygen levels are higher than at the seabed. And just before the sunrise, they return to the bottom and hide. We looked into fisheries data at the Walvis Bay in Namibia, 15 years data. And they show that gobies join layers with jellyfish when they swim up in the pelagic, and are 6 times more likely to be caught with jellies than their predators are. So the gobies can then avoid being eaten. Yes, they are specialists in hide and seek, which is made possible by extreme physiological and behavioral adaptations. And experiments show that jelly appears to be a safe refuge. And horse mackerel strongly avoided jellyfish. Horse mackerel is a predator on the gobies, while gobies did not avoid to swim with jellies in the experiments on board GO2NE. Aquaria experiments also show that mud is the preferred substrate by the gobies. And stable isotope analysis and also stomach content analysis have shown that they have an unusual menu. Jellies, the diet consisted of 60% of jellies and 35% of the sulfide-rich diatomaceous mud. We also had physiology experiments that show that hake hearts did not recover after they were exposed to anoxia, while the goby hearts, they did fine. So the goby seems to be able to hold their breath for hours. And both the respirometry and aquaria experiments show that they pick up oxygen from the surroundings better than any other marine vertebrate. And they can hold their breaths for hours if oxygen is absent. So why have the gobies been successful on the hypoxic Benguela chapter? Well, just a little repetition. The reason for its great success is that they are specialists in hide and seek, made possible by the extreme physiological and behavioral adaptations. Oh, a little food competition. They have a very special diet. So we may interpret this as gobies could be rebalancing the food chain. They are able to cope with this hypoxic and sulfidic-rich environment that has been in the Benguela shelf for years, for many hundreds of years. And by feeding on the dead end products, such as jelly and mud, they bring back otherwise lost resources to the food chain and the fishery in the Benguela ecosystem. And they increase in abundance when the hypoxia is severe and predators escape. And they also reproduce on shelf edge and allow upwelling currents transport larvae to a suitable nursery area. And then perhaps stabilizing the production in the Namibian Benguela. So that was the end, and thank you to all my collaborators. I don't have all the pictures of them. There are so many students and colleagues in South Africa and Namibia and Norway. So thank you.
Thank you, Anna Groh, for that excellent and fascinating presentation. I know how much work must have gone into that. You make it look so simple, but I would encourage our listeners to put questions in the Q&A. There aren't any right now, so I'm going to ask you one, and so we have some time for them to come in. I am wondering, this is such a fascinating fish with so many interesting adaptations. Are there any analogs to this fish in other parts of the world where oxygen is very low? And, would you expect sort of our future warmer, less oxygenated ocean to favor such fish, not just gobies, but is there anything else that has remotely the capabilities of the goby to tolerate low oxygen and maybe hydrogen sulfide?
I think there must be gobies other places in the upwelling ecosystems. I expect there are. I haven't read the literature yet about it, but I do expect that when we get, if the shelf areas will increase, or when climate changes, if the shelf area with the hypoxic environments do increase, then we also would expect that there will be more favorable for species that tolerate, that are adapted to this lower. To this extreme environment, and the gobies, they must have evolved for thousands of years in the Benguela. It's an old shelf system, and this species is endemic to this region, and I expect there are similar analogs in the other upwelling ecosystems that also have been there for thousands of years, or maybe millions of years. I don't know. Yeah.
Okay, great. Thank you. The questions are starting to come in. So we have a question from Nancy Rabelais. She asks, what are the physiological adaptations other than breath holding? And then she asked maybe blood chemistry?
Well, I was not we did collaborate with physiologists to do that work, and I'm not very much into the details about the physiology. So I'm sorry about that. It's a very good question, but there are more than breath holding. Yes, there are.
Well, the heart, I guess you showed us the heart.
The heart, yeah, the heart shows they did fine after the physiological experiment when we exposed the hearts for anoxia and hydrogen H2S as well. So yeah, and they did gobies did fine, but the hakes, they just passed away. Yeah. So.
Okay, we have a question from Beatrice Scotto. She says, thanks for your interesting presentation. I was wondering if the results of the field campaigns have been published in a database to be viewed.
We have published the papers, but we have not sent the data into any database. No, yeah, sorry. But if other people want to look into them, they should contact us.
Okay, good. There's a question from Carlos Niera. He asks, is there evidence that these gobies feed on bacterial mats? Based on your pictures, it looks to be Thioploca or Beggiatoa mats that are present.
Yeah, it was these Beggiatoa mats they were feeding on, and also on the jellies. But we don't know whether they feed on the jellies while they are hanging up in the water column, or if they feed on the dead jellies on the seedbed that has been done at the seedbed. Yeah.
Okay, thank you. We have a question from Amid Mohammadpour. He says, I appreciate our informative discussion. Considering the goby's fish remarkable tolerance to low oxygen levels, could it serve as a viable option for widespread cultivation in fish farms across various temperate regions globally? Thank you.
So to breed gobies in fish farms, well, it is possible. I think it could be possible to breed them, but I think there are. I think the predators that eat those gobies have a higher value than the gobies would have for nutrition, for food and for nutrition. Yeah.
Okay, let's see. We have a question from Anders Tengberg, who writes, we had an observatory in a frequently anoxic Swedish fjord. The acoustics show daily migrations of zooplanktons down into hypoxia. Can zooplankton handle anoxia and hypoxia? It's not
I think
specific to
there,
the goby.
well, I think some species might handle low oxygen, and what we find in the Norwegian fjords is that we find krill even deep in areas where oxygen levels have been very low. So that in areas where that has had low oxygen levels for a long time, I expect some animals to be adapted to that.
So there's a question from Natalya who can't put it in the Q&A because she's a panelist, but I'm going to ask it for her. She asks, I'm curious if closely related species to the Benguela goby also show high hypoxia tolerance.
Closely related species would then be from the goby family, I presume. Yeah. So I am not familiar with any other goby, but I expect there will be some. Maybe other people know about that.
I mean, I know gobies occur in tide pools that sometimes can go into low oxygen, you know, in the intertidal, but
Yeah.
I don't know as a group if they're known to be especially hypoxia tolerant. If anybody, and if any of our listeners know that, they could.
Yeah. Maybe some other listeners know, but the goby population of Namibia is, to my knowledge, that's the biggest population with this kind of adaptation when it comes to a goby. Yeah. But there might be a lot that I don't know of.
Okay. I don't see any other questions. There were a couple of more questions for Natalya, but they've disappeared off the screen. And we still do have a few minutes if anybody wants to ask their question again and didn't get them answered from the first presentation, or if anybody has additional questions for Anna Groh, you can put them in. It's been a fascinating look into the responses of fish to low oxygen. I think one of the things that's come out, it seems like from these presentations, is that and you know, a really good takeaway is we think of fish as being rather intolerant of low oxygen, but it's clear both of Namibia and California that there is lots of tolerance. So Natalya, would you like to add anything to that?
Yeah, I agree with that comment, Lisa. I think every single low oxygen system in the world you can look at, you'll probably find a demersal fish species that has evolved to be able to tolerate those extreme conditions. We wrote a review paper on this looking at oxygen minimum zone. So as long as it's a system where those conditions have been present through time, we find demersal fish that have adapted to those conditions. So fish do show remarkable tolerance to low oxygen conditions, but you know, a very small proportion of the overall species diversity in the world have that really high hypoxia tolerance.
Okay. Would anybody else in the audience like to weigh in with question or comment? And you would have to put it in the Q&A. Otherwise, let's see. There's one from Nancy. Let me just read what she says. She says, in the northern Gulf of Mexico, gelatinous zooplankton will go into the low oxygen, but vertical migration is disrupted. Studies by Mike Roman of the GO2NE steering committee, I think, have also shown this. So that was probably in response to the question about whether zooplankton can migrate into low oxygen waters. So thank you, Nancy. Any other thoughts or comments? And Anna Groh or Natalya? If not, I will wrap up and let me share my screen for 1 second. I do have a final slide.
I have a comment, Lisa.
Yeah. Oh, go ahead.
Yeah. I guess I would just like to leave the audience with a comment that we should also think about trophic interactions when thinking about changes to species distribution with hypoxia, and that it's probably, at least the changes that I saw in my study in the Southern California bite, it's likely interaction of both kind of the challenging physiology of the hypoxic environment for the demersal fish, so that affecting the fundamental niche, right, of the fish, its actual physiological tolerance, but then also the realized niche being a matter of the availability of the prey that it can prey on. And so I think a lot of times when we think about changes in species distribution with hypoxia, most of the time we're thinking about that kind of physiological tolerance piece. But I think that the changes in prey availability and those trophic relationships is an additional important point for us to think about as we consider deoxygenation impacts. And I think Anna Gro's example too is a really good one, showing this really hypoxia-tolerant species being one that plays a key role in this bentho-pelagic coupling process off of Namibia. So I think there are cool trophic interactions to explore in looking at deoxygenation impacts.
Thank you for that, making that really insightful and important point. And with that, I'd like to thank all our listeners for their active participation, and we look forward to welcoming you again for the next webinar in October. And also, please check out the good news for the latest news and events in the ocean deoxygenation community. So thank you all, and I wish you all a pleasant day or night from wherever you are connected.